Okay, welcome to video number 16 of the Diaries of a Coronavirologist YouTube channel. Today is the 22nd of April, and we're up to about 2.6 million confirmed cases of COVID-19 and about 183,000 deaths. Sorry that I haven't posted for a little while. I've been preparing two much larger presentations, and that's also going to mean I don't post as frequently for about a week or so. I'm doing presentations on Monday and Thursday next week. Now, these are much longer form presentations that I'm having to prepare and put a lot of time into perfecting, and that's taking up a lot of the time that I put into doing these shorter videos. I'm hoping, however, that they may be available for people to check into live or potentially be recorded and put online after the fact. So if that happens, I will post them here or link to them here and link to them on my Twitter feed. So for those who don't already follow me on Twitter, I always post my handle down in the description so you can find it there, and I'll try and keep an update on whether those presentations are available. If they're not made available after the fact, what I may do is re-record myself doing them for anyone who's interested in the longer form of a scientific presentation. A lot of the stuff I'll talk about has actually been covered on this channel already, but it's just putting it all together and practicing it and trying to perfect it before I give those talks. That's going to take up a lot of the time and mean I post a couple fewer videos over the next week or so. So just to do a bit of housekeeping and give you a heads up of I may not post so many videos over the coming few days. What I want to talk about tonight is antibodies and how they can inhibit virus infection. So this is building on my last video where I was talking about vaccines. I wanted to go into a little bit more detail about why antibodies work, or how antibodies work, and how they can inhibit virus infection. And at the same time, do a little bit of virology 101 and talk about how viruses infect cells, because how viruses infect cells is how antibodies are capable of inhibiting virus infection. So viruses can only infect certain types of cells, and this is something broadly known as tropism. So a virus's tropism is the certain types of cells that it can infect. One of the major factors of tropism is the receptor, and whether a cell has the receptor on its surface that a virus can bind to and use to infect that cell. So I'm going to keep using an analogy that I've used on this channel before, and one that's very often used when talking about viral infection of cells and receptors, which is a lock and key. So the receptor is the lock and the virus has a key. So the key on the virus are a group of proteins that are referred to as glycoproteins. So if we think of coronaviruses, again, something I've done on this channel before, the bits that stick out of the surface of the virus, the spiky bits, for coronaviruses, they're called spike, but more generally, they're called glycoproteins. To give other examples, for influenza, the glycoprotein that's involved with entry is called hemagglutinin. When you think of the way we talk about influenza viruses, you may be aware of things like H1N1 or H5N1 or H7N9 and so on and so forth. The H is hemagglutinin, and the numbers denote different types of hemagglutinin protein. The N stands for neuraminidase, just for anyone wondering, and that's more involved in how the virus gets out of a cell, but that's a very different topic. For a virus like HIV, which I'll use again as another example shortly, the glycoprotein involved with entry is called envelope. So different viruses have different glycoproteins, and that determines which cells they can infect. Again, I'm going to keep using this lock and key analogy. So the receptor that is used by the SARS2 coronavirus, that is the virus causing COVID-19, is called ACE2. This is angiotensin converting enzyme 2. It's the same protein that is used by SARS1, so I can talk about the two of those in the same breath in a way. To take the other major pathogenic human coronavirus of MERS coronavirus, this uses a different receptor which is called DPP4, dipeptidyl peptidase 4. So those three viruses, SARS1, SARS2, and MERS, have different tropism because of the fact that certain cells express ACE2 that don't express DPP4, and there's certain cells that express DPP4 that don't express ACE2. So certain cells have the correct lock for SARS coronaviruses to unlock, and certain cells have the correct lock that can be unlocked by MERS coronavirus, to keep coming back to that analogy. So why knowing the receptor is important is it allows us to understand how these viruses spread. We know that cells in the lungs express ACE2 and DPP4, so we therefore can speculate and obviously experimentally prove that coronaviruses spread through respiratory transmission. With HIV, for example, as I said I'd come back to, the receptor is a protein called CD4, which is found on white blood cells, for example, but is not found on cells in the lungs. So you can't breathe in HIV and get infected by it in the lungs as you could with coronaviruses. So this is just some of the really basic virology about how certain viruses are capable of infecting certain cells and how that can play a role in how they spread and the certain types of diseases that they can cause. And I wanted to go over this because I want this YouTube channel to be educational, and also it's really important for how antibodies work and how they can protect you from infection. So to bring things together, the way antibodies work and what we're trying to design vaccines to produce antibodies to do is to inhibit the ability of a viral glycoprotein to interact with a receptor in a productive way. So there's two ways that can work, which is either you block the ability of the key to fit into the door, or you block the ability to turn that key in the door. So in a general scenario where a virus infects a cell, the spike protein can fit perfectly into its receptor, and that allows for infection. A big old antibody sits on top of that spike. It can't interact with its receptor. With 3 hands, I could demonstrate that a little bit better, but I think you get the point. There's more detail I could go into, and I probably will in a future video, about how that turning of the key in the lock actually allows for infection. But that's another way that antibodies can inhibit, is by blocking the turning of the key in the lock. So this was just a quick video I wanted to put out because I could kind of do it largely off the top of my head without checking too much about the most recent research that's been going on. I want this channel to be both broadly educational about how viruses infect cells, as well as very directly relevant and up to date about SARS2. I know people have been commenting and asking questions about things along the lines of how long the virus can survive on surfaces, how long it can stay in the air, is it airborne versus respiratory droplet transmitted, and all these kind of things. And I do want to get to those videos. Just right now, because of preparing these other presentations I mentioned at the top, I'm a little bit strapped for time, so I'm going to fall back on some of the more general virology things, just to keep putting videos out in the meantime before I can go and read all of the most up-to-date literature and convey that to everyone through the channel. So as always, please keep commenting and leaving feedback. I may not respond on the channel, but I am trying to keep on top of looking at it and therefore directing which videos I'm going to put out, what topics I'm going to talk about in future. And if you're finding these videos valuable, please keep subscribing and sharing and dropping likes on them and all those kind of YouTube things. And as always, please stay safe, wash your hands, and keep calm and carry on. We will get through this pandemic. Thanks for watching.